All right. Hello, everyone. Today we have a special webinar as part of our Let's Talk NMD, Let's Talk Neuromuscular Disorder series. Today we have Dr. Toshi Yokota here today to talk about FSHD 101. And we decided to do this webinar in honor and in celebration of World FSHD, which is on June 20th. Every June 20th, people around the world join in activities to raise awareness for FSHD. And this year, we wanted to give a chance for individuals to learn a little bit more about FSHD, brush up their knowledge on FSHD, and have an opportunity to ask questions about FSHD. It's wonderful that we have one of the Canadian researchers looking at FSHD on this webinar today. Dr. Yokota's research focuses on understanding and developing treatments for muscular dystrophies and other genetic disorders. In particular, his research aims to develop and apply new methods, including precision health and personalized genetic medicine, to correct mutations in a precise and targeted manner. We're really grateful that we've got Dr. Yokota. He is a professor at the University of Alberta, the Friends of Garrett Cumming Research Chair in conjunction with Muscular Dystrophy Canada, and he is an MDC MSAC member, Medical and Scientific Advisory Committee. He's also an HM2PEN Neurological Science Research Chair. So I'll turn it over to you, Dr. Yokota, to start us off on some information around clinical and research updates when it comes to FSHD.
So can you hear me now?
Sounds great. Go ahead.
Yeah, sorry about that. So thank you for your introduction, Homira, and thank you for having me here today. I'm Toshi Yokota from the University of Alberta, and I'm Muscular Dystrophy Research Chair. And today I will talk about 101 FSHD. So today I will cover several topics. First, I will talk about the basics of FSHD, and I will talk about symptoms and the signs of FSHD, and then I will talk about how FSHD is diagnosed and the prognosis, and also I will talk about treatment options and supportive treatment for FSHD. And lastly, I will talk about research going on in Canada on FSHD, and mostly I will talk about research in my lab. Okay, so the first topic is what is FSHD? So FSHD stand for Facioscapulohumeral muscular dystrophy. So it's a bit mouthful. It's a bit mouthful, but it means face and shoulder and upper arms. So basically, FSHD affects these muscles mainly. And this is the third most common form of muscular dystrophy. 1 in 8,000 to 22,000 people are affected. And this is a genetic disease. And a child who has a parent with the mutated gene has 50% chance of inheriting FSHD. So it's called autosomal dominant disease. And approximately 30% of the individuals with FSHD have no apparent family history. So basically everyone has chance to have FSHD. And it's progressive. It's quite slowly progressive, but it's progressive disease, and it's spread to the body-wide muscles. And thus, rare infantile form also exists. I will talk about this more in detail. And next, so this slide shows brief history of FSHD research. So basically, disease itself was first distinguished as a disease in 1870s, so it's quite old. But research progress has been quite slow, actually. That's because FSHD is such a complicated disorder. So the DAX4 gene, this is a disease-causing gene of FSHD, and that was discovered in 1999. But at that time, it was not known that DAX4 was the disease-causing gene of FSHD. So it's not identified as an FSHD disease-causing gene. So in 2007, DAX4 was found to be expressed and toxic. So it's actually produced in the body, and it's also have toxic effect in patients. And in 2010, genetic mechanism causing DAX4 expression identified. So in 2010, so it's just 10 years ago, disease mechanisms were fully understood. And also, it's not fully understood, but at least DAX4 was identified as disease-causing gene of FSHD. And in 2012, FSHD2 identification of predominant mutation of FSHD2 was discovered. And then in 2019, first clinical trial to reduce DAX4 started. So research progress has been quite slow, but that's because FSHD is really complex disease. But since causative gene was identified, research has significantly progressed. So yeah, as I mentioned, FSHD is such complex disease. So it's basically overexpression, so production of toxic protein called DAX4 in the body. So it appears seemingly simple, but inside was quite complex. So it's like a layered cake inside. So yeah, be prepared. I will talk about this. Yeah, so FSHD is basically caused by DAX4 misexpression. That means production of DAX4 protein in the body. So basically DAX4, everyone has a DAX4 at very beginning in the developmental stage. So in 4-cell stage, so when we have only 4 cells, DAX4 is expressed in these cells. Everyone has DAX4, but then it's turned off, and in adult tissues and cells, healthy people don't have DAX4 gene expression. So no DAX4 production, except for testis and thymus. But in FSHD patients, it's for some reason turned on. And this DAX4 protein has very toxic effect, especially on muscles. So that causes FSHD. So yeah, this slide shows simple mechanism of FSHD1 and FSHD2. So there are two types of FSHD. One is called FSHD type 1, and another one is called FSHD type 2. And but both involves DAX4 overexpression, production of DAX4. And DAX4 overexpression, production, basically leads to muscle atrophy. So muscles become thinner, and it causes massive inflammation in the body. And also it disrupts myogenesis and muscle differentiation. So muscle fibers, muscle cells cannot grow and cannot become thick and fully mature. And also it causes glutathione redox pathway. So this causes oxidative stress. So it damages muscle cells. Okay, so sorry. So next slide shows more detailed downstream pathway in FSHD. So it's becoming quite complex. But you don't have to memorize these signaling pathways. But basically, DAX4 expression, gene expression in muscle fibers activate lots of downstream genes and pathways, and that leads to muscle atrophy and disruption of immune systems and abnormal muscle cell formation and oxidative stress and muscle inflammation. So all of these contribute to the FSHD pathogenesis. So this slide shows how FSHD1 is caused. So if you look at DNA, the region of DAX4 gene, and you compare normal healthy individuals and FSHD patients, healthy individuals have more repeat of the DAX4 gene. So it's a bit counterintuitive. So healthy people have more repeats, and patients have less repeats. The thing is, in healthy individuals, if there are many 11 or more repeats, DNA is tightly coiled and the gene is can't expressed, can't read. So DAX4 is not expressed. But if the repeat sequence is fewer than 11, the DNA coil in this region is more relaxed, and the message can be read, and that leads to production of DAX4 expression, and it leads to FSHD symptoms. So that's the basic mechanism of FSHD type 1. Okay, so yeah, as I mentioned, there are two types of FSHD, and the mechanism is a bit different. And as I mentioned, healthy individuals have 11 to 100 repeats of um, repeat sequence. It's called D4Z4. So basically, D4Z4 consists of 11 to more than 100 repeat sequence segments, and each of them have about 3,300 DNA base pairs. So it's quite a long DNA repeat sequence. And in FSHD patients, they have less than 10, 10 or less repeats, and that leads to DAX4 expression. And in FSHD2 patients, they have basically, they usually have 12 to 16 repeats, but they have mutations in other epigenetic genes. That means these genes are involved in the relaxation of the DNA in this region. So that leads to DAX4 messenger expression and production of DAX4 protein. So that's how DAX, FSHD1 and FSHD2 patients have DAX4 expression. So, but both involves DAX4 expression basically. Okay, so next part I will talk about symptoms and signs of FSHD. So symptoms, as I mentioned, face muscle and shoulder muscle and arm muscles are three most affected muscles in patients. So they have symptoms typically like inability to whistle, inability to sip through a straw, and eyes that don't close fully during sleep, and difficulty with sit-ups and pull-ups, shoulder blades that wing out like this picture, and difficulty raising arm beyond above shoulder height, and weakness in hands and fingers. So that's a typical symptoms of FSHD patient. And some patients have hearing complications in severe cases, and they also sometimes have foot drop and trouble walking. And sometimes they have big abdomen, and that causes constipation as well. Okay, so this slide shows the muscles affected by FSHD. So face muscle and shoulder muscle and arm muscles are most affected, but some patients have weakness in abdominal muscles and tibialis anterior muscles as well. Okay, so next topic is diagnosis and prognosis of FSHD. Okay, so this flow chart shows how FSHD is diagnosed. So as I mentioned, it's genetic disease. So many patients, about 70% of the patients, they have genetically confirmed first degree relatives. So in that case, they likely have FSHD. And otherwise, they test for presence of D4Z4 contraction. So this is, as I mentioned, it's DNA testing basically. And if that D4Z4 contraction is detected, and they have clinically typical FSHD, then they will likely have FSHD1. And also they can test clinicians, and they can test for A allele on contracted D4Z4. So this is a bit complicated, but there are two types of in chromosomes in the region in the DAX4 or encoding the chromosome, and only type A allele. So chromosome type A can lead to FSHD1. So if they have A allele, then they will likely have FSHD1. And otherwise, they will likely have other myopathies. So yeah, so there are several si- several diseases with similar symptoms. So um, to distinguish with these diseases are very important. Okay, so this slide shows prognosis of FSHD. So onset is quite variable. Some patients, about 4% have infantile form. So some patients have start symptoms as early as 3 years old, and some patients start at 72 years old. So it's quite variable. But about 90% of patients have developed symptoms before age 20, so when they are teenagers. And it's progressive disease. Initially, FSHD affects upper portion of the body, but a lower portion gets affected over time. And about 20% of patients become wheelchair bound. Um, but the good news is FSHD doesn't typically shorten the lifespan. Most patients live normal lifespan. Okay, so next part is treatment options and supportive treatments for FSHD. So in this figure, left side, left panel shows support and treatment for infantile form of FSHD patients. So diagnosis and the genetic counseling service are available, and also physiotherapy and speech rehabilitation and orthopedic scapular spinal correction. And it's not available for everyone. This surgery can keep the shoulder blade in one spot to help your child use his or her arms, but it's not always successful. So it's not available for everyone. And also epilepsy and pain control and hearing aids, otological monitoring. So hearing hearing impairment is also um, symptoms in some patients, and pulmonary care, sleep study, and ophthalmology monitoring. And there are novel disease modifying drugs, clinical trials as well. So these are available options. But unfortunately, there's no available cure for FSHD. But researchers, including myself, are working hard to develop new therapy for FSHD. Um, so mild and low intensity exercises are available. Occupational therapist or physical therapist can plan safe program, and non-steroidal anti-inflammatory medicines are available to ease muscle inflammation and pain. And as mentioned, scapular fixation is an option for some patients. And supportive devices like corset, back support, bras, lower leg braces, and high top shoes are also available. Okay, so in the final part, I will talk about research being conducted in Canada on FSHD. So currently, Muscular Dystrophy Canada support a couple of research programs on FSHD. So one is Dr. Rima Al-Awera in Ontario Institute for Cancer Research on safety and efficacy of possible epigenetic therapy for facioscapulohumeral muscular dystrophy. And the other is our research on enhancing the efficacy of antisense oligonucleotide therapy for facioscapulohumeral muscular dystrophy. Um, so today I will talk about research ongoing in my lab. I cannot talk about Dr. Rima Alawia's research, so I will mostly talk about my research. Okay, so in our lab, we are working on the development of new therapy for FSHD. And there are several or almost 10 different strategies to stop DAX4 activity. And for example, um, targeting gene repression with CRISPR-Cas9 and CRAB. This is called genome editing to stop the production of DAX4. And also correction of SMCHD1 mutation. This is for FSHD type 2 patients using genome editing as well. And there are strategies for gene knockdown with antisense oligonucleotides. So antisense oligonucleotides are basically DNA-like molecules, artificial DNA-like molecules to stop the production of DAX4 by binding the gene transcripts of DAX4. And also, there are gene knockdown and silencing strategy with RNA interference. So this is also strategy to stop the gene production, but this is by using siRNA or RNA interference. So this is not antisense artificial oligonucleotide, but instead RNA, artificial RNA can be used. So DNA aptamers, these are DNA molecule that can bind to DAX4 protein to stop the activity of DAX4. And introduction of protein homologous or similar to DAX4 or DNA decoy. So basically, these strategies all target DAX4 protein and stop the activity. And also, gene knockdown and silencing of DAX4 downstream targets are also important strategy. So even if you cannot stop DAX4 activity, you can maybe stop the activity of DAX4 downstream genes to stop the symptoms. And next one is gene knockdown and silencing of genetic modifiers. So we can target other genes that can affect FSHD symptoms. And also we can deliver genes coding for proteins that ameliorate DAX4 mediated toxicity or secondary feature of FSHD pathology. So gene therapy can introduce gene that can ameliorate FSHD symptoms. So these are all strategies currently ongoing in the labs. Yeah, so in our lab, basically we focus on gene knockdown with antisense oligonucleotides using DNA-like molecules. Okay, so this slide shows the basic structure of antisense oligonucleotides. So as I mentioned, this is DNA-like molecules, and antisense oligonucleotide therapy basically means an approach to fighting diseases using DNA-like molecules. So if you compare these molecules, for example, LNA or 2' MOE, these are examples of antisense oligonucleotides, and these have quite similar structure to RNA or DNA, but only slightly modified. So these modifications make these oligonucleotides less toxic and more effective. Okay, so yeah, to introduce to get into more detail, I will just talk about a little bit about the basics in genetics and protein synthesis. So this slide shows how DAX4 protein is synthesized from DNA. So in DNA, basically there are many regions called exons. These exons basically carry information that's to produce DAX4 protein. And introns are other regions. So after transcription, DNAs are transcribed and messenger RNA are synthesized. Then messenger RNAs are spliced together. So only exons are connected together and they make mature messenger RNA. So mature messenger RNA only have exons. And then mature messenger RNAs are exported from nucleus to cytosol outside of nucleus, and then it's translated and proteins are produced. So basically, our antisense oligonucleotide technology target messenger RNA, and antisense oligonucleotides can specifically bind messenger RNA that have information to produce DAX4 gene expression. So antisense oligonucleotides are very specific. So it specifically target DAX4 gene. Other genes are not affected. And antisense oligonucleotides can introduce RNSH. So this is enzyme that can degrade messenger RNA. Then RNSH can degrade and destroy messenger RNA. So the cell no longer translate and produce DAX4 protein. So that's how antisense oligonucleotide works. Okay, so our antisense oligo is called Gapmers. So Gapmers basically means um, it's kind of hybrid structure of DNA and modified antisense oligos. So basically, central part has DNA. So it's natural DNA. It's artificially synthesized, but the structure-wise, it's the same as DNA structure. And we can use modified chemistry as both end of Gapmers. And these modified antisense oligos can strongly bind to DAX4 RNA. And then central DNA part can introduce RNSH. And then RNSH can degrade and destroy messenger RNA of DAX4. So that's how antisense oligos called Gapmers work for DAX4. So we recently published a couple of papers on this. We can inhibit DAX4 expression using antisense oligos called LNA and Gapmers as therapy for FSHD. We published this last year in PNAS proceedings of the National Academy of Sciences of the United States of America. And the other one is DAX4 knockdown with antisense oligo. We use 2' MOE antisense oligo Gapmers. So both LNA Gapmer and 2' MOE Gapmers worked very well in cell model and mouse models. So yeah, if you are interested in these papers, please, these are open access papers. So please refer to these papers. So basically in our studies, we tested the effect of antisense oligonucleotide Gapmers, and we used two models. One is cell model, patient-derived muscle cells, and the other is mouse model. So FSHD mouse that express DAX4. And we designed several antisense oligonucleotides. So as I mentioned, there are several exons in the gene that carry the information to produce protein. And we targeted this specific region because of the several technical restrictions. And Dr. Echigoya and Dr. Maruyama are involved in the design of these antisense oligonucleotides. And we use 7 antisense oligonucleotides of LNA and 3 antisense oligonucleotides of 2' MOE. And sorry, we use 7 antisense oligo for 2' MOE. So LNA. Okay, so so at first we tested these Gapmers in the cell model. So we can use patient-derived cells to test antisense oligo Gapmers. So the thing is, DAX4 is only produced in very fully mature myotubes. So mature and differentiated muscle fibers. So basically, what we did is we culture cells of muscles. It's called myoblasts, and they fuse together. So basically, mature muscle cells have many nuclei in the cell, and we grow after 7 to 18 days. FSHD patient cells produce DAX4. So we can test the effect of antisense oligonucleotides in at 10 to 18 days after the cell culture differentiation. So we tested several antisense oligonucleotides, LNA and 2' MOE, and they worked very well. Almost 100% reduction of DAX4 expression was observed after single transfection. So that's great. And also several downstream genes. So ZSCAN4, TRIM43, and MBD3L2, these are downstream pathway of DAX4. So basically, if DAX4 is produced, these downstream genes are also upregulated. So after the knockdown of DAX4, we can also knock down these downstream genes using LNA and 2' memory Gapmers. So that can ensure that these antisense oligos can knock down not just DAX4, but all downstream pathways involved in the FSHD. Okay, so we also looked at the morphology of cells after the treatment. So in healthy muscle cells, muscle fibers are very thick and they have many nuclei. So basically, these blue signals represent nuclei, and there are many nuclei in healthy muscle cells. But in FSHD patient cells, these cells look obviously smaller, thinner, and also many of them have only one nucleus. So they cannot fuse together and they cannot grow very well. They cannot um, become very thick. So after treatment with antisense oligonucleotide Gapmer, we confirm that cells can fuse together better, and also they become thicker. So, so this is very promising. Myotube fusion and myotube diameter both are improved after the treatment. So this graph shows average fusion index of myotubes. So after the treatment, myotubes, muscle cells can fuse very well. So that means they can differentiate and develop very well. And also number of, so the size of myotubes are shifted toward healthy cells. So this basically graph shows myotube diameter and number of myotubes. So in non-treated cells, many cells have very small diameter, narrow diameter. But after treatment, these cells become thicker. So the morphology is also improved. And the same tendency was observed in myotube diameters. And also both LNA and 2' memory Gapmers worked very well. Okay, so next part was done in FSHD mouse model. And this part was done by Dr. Yuan Chen's group in Children's National Medical Center. And the mouse model was developed by Dr. Peter and Takako Jones group in University of Nevada, Reno. And we tested antisense oligonucleotide in mouse model of FSHD. And we looked at the reduction of DAX4 expression after the treatment. So, so it worked very well. Both LNA and 2' memory significantly reduced DAX4 expression, and 2' memory also reduced DAX4 expression. So this is just after a single intramuscular injection. So, so far, it looks very promising, and we are currently working on systemic treatment. So basically, LNA and 2' Gapmers both effectively knock down DAX4 expression, and systemic treatment studies ongoing. We are looking at both safety and efficacy in FSHD mouse model. And we are also testing new Gapmer chemistries to make them more effective and less toxic. And we are also testing new delivery systems so that deliver better to body-wide skeletal muscles. Okay, so that's about it. And most of this work in my lab was done by Kenji Lim. He's PhD student, and he just graduated earlier this month. And these are our lab members, and Dr. Yuan Chen's group is working on a mouse study of antisense oligos. And Takako Jones and Peter Jones provided mouse model, and Dr. Charles Emerson and Jennifer Chen provided a cell model. Okay, so and these are funding agency and Muscular Dystrophy Canada and FSHD Society and FSHD Canada Foundation, CIHR, and Friends of Garrett Cumming Research Fund, and many other funding agencies supported this study. So I'd like to thank these funding agencies. Okay, thank you very much for your attention.
Thank you very much, Dr. Yokota. That was a wonderful overview of FSHD and then an opportunity for us to learn about the most recent clinical and research updates. On behalf of MDC, I actually want to congratulate you. As you mentioned, you recently have been awarded a grant through
Thank
a
you.
research competition looking at FSHD. And as you mentioned, this year we also have granted another award. It's a Team Science International grant together with CIHR through the European Joint Rare Disease Program. We have funded an FSHD study. And I'm just going to quickly read what that study is all about. They've noted here that FSHD is caused by gain of expression of the double hemiobox 4, DUX4 gene, causing toxic effects in muscle cells. Despite the consensus on the pivotal role of DUX4 in several clinical trials, there's currently no cure or effective approach for therapeutic approach for FSHD. So in their studies, they're identifying a novel regulator of DUX4 expression. Targeting this factor allows to block DUX4 expression and rescues the pathogenic behavior of muscle cells from FSHD patients. They're noting that the treatment is safe to healthy muscle cells, and based on their results, they would use a cellular and animal models of disease to investigate a novel pharmacological approach that could represent a promising therapeutic option for FSHD patients. So this is great in combination with the work that you're doing. I think together we're learning more about FSHD. These are just two examples of research that MDC has funded in FSHD. We've funded other research studies as well, whether they're directly studying FSHD, and then some that are indirectly impacting the lives of Canadians affected by FSHD, like with the Neuromuscular Disease Network for Canada. So that is great. I wanted to take a moment just to ask a couple of questions that have come through our research hotline. And just for those who don't know about our research hotline, if you have any questions about your disorder, you can email research@muscle.ca. One question that has come in is you mentioned that FSHD can affect, multiple, it's multisystemic in some ways, right? It can even contribute to hearing loss, as you mentioned. An adult affected by FSHD is talking about having quite a bit of challenge in terms of accessing multidisciplinary support. Have you heard some good models around multidisciplinary care teams for FSHD in adults?
Care teams. That question is quite clinical, and I'm a basic researcher, so I'm not very familiar with the support team, clinical care, but there are many muscular dystrophy you know caring facilities in Canada, and yeah, so I and muscular dystrophy Canada can I think recommend these
Yeah, absolutely. Maybe,
facilities.
yeah, and I can add that to that question too. I wanted to really highlight it, even though I know we're bearing in mind your research and expertise. I want to highlight it because I think it's a common experience for many adults
Right.
affected by neuromuscular disorders where, at times they have really comprehensive care in pediatrics and as youth, and then you transition to adulthood and you're on your own, so to speak, to find a PT, a speech language pathologist, an audiologist. But yes, please contact your service specialist and they will help to be able to navigate, find you an appropriate individual, a multidisciplinary care team that can help to address your needs. So I wanted to go to that question. The next question is, Dr. Yokota, in your opinion, how close are we to a treatment becoming available for those affected by FSH?
Yeah, that's a very, very tough question actually. Yeah, our group is developing antisense therapy, and we hope to start clinical trials as soon as possible. Maybe next few years or so. And many groups in the world are developing antisense or other therapeutic strategies. And so because I think the important part is the disease mechanisms of FSHD was identified quite recently. So, uh, you know, basic mechanism of FSHD was identified just 10 years ago. So, um, so this um, this research area is quite new. It's still in its infancy, but the research has, I think, quite rapidly developed recently, and lots of very encouraging and promising research are ongoing. So I think in next few years or several years, we will see lot more clinical trials starting all over the world.
Another question that we had is, what about therapeutic approaches for pain management? So we have an adult here in their mid 20s mentioning that they have severe pain in his shoulders, and was wondering, well, what about if we don't have a cure for FSHD, and we might be a few years off from therapeutic approaches for FSHD, are there some management in terms of, or therapeutic management areas that you want to talk about around pain for FSHD?
Yeah, I hear lots of patients complain about pain in FSHD, and so yeah, basically as I mentioned, non-steroidal anti-inflammatory medicines used to ease muscle inflammation and pain. So basically this is the option for FSHD patients, and I think you can discuss it with your doctor and look at the anti-inflammatory medicines.
Excellent, thank you. And then another question here is, you've mentioned antisense today as part of therapeutic approaches, and they've heard of this term with other neuromuscular disorders. What can we learn from other neuromuscular disorders and apply it to FSHD as it relates to antisense oligonucleotides?
Yes, that's great question. So antisense oligonucleotide therapy has been approved in a couple of neuromuscular diseases, DMD and Duchenne muscular dystrophy and spinal muscular atrophy, and so this is I think very encouraging antisense, especially for DMD, because in DMD antisense oligos are targeted for body-wide muscles as well and heart. But in SMA, antisense oligos are used basically for motor neurons. But in FSHD, we also want to treat body-wide muscles. So the success of antisense oligotherapy for DMD is especially very encouraging. And so basically, antisense oligo can work very well for muscles. And the mechanism is slightly different. For DMD, antisense oligo can restore the gene expression and increase the dystrophin protein. But basically, what we are doing for DAX4 is opposite. So we want to knock down and to reduce DAX4 expression. So the strategy is opposite, but basically we can deliver antisense oligos at least to body-wide muscles. So that's very encouraging, and hopefully antisense oligonucleotide therapy clinical trials will be started in next few years or several years, hopefully.
Very good. Another individual has sent a question saying that they recently, their genetic testing results indicated mutations in the TTN and PLEC genes. And the question is that without having testing, how's that helpful? Should people continue to receive genetic testing? Is that helpful in the case of FSHD?
Sorry, the connection was a bit interrupted, but but basically the question is, antisense therapy can work in other muscular dystrophies.
Yeah, I can
Yeah.
go ahead and repeat it. The question is, an individual has mutations recently revealed in terms of the genes TTN and PLEC. And their question is that without therapeutic approaches currently available for FSHD, is genetic testing important?
Yes, genetic testing is very important, especially for for example, titine deficiency. exon skipping strategy used in DMD can work for tightening as well. And some researchers are working on the um, exon skipping therapy for tightening. So genetic testing is very important, and patient registry programs have started in many countries, and that's very helpful to figure out how many patients we have who are amenable to exon skipping and antisense therapy. And so, yeah, one important aspect in FSHD is basically antisense therapy can work for almost all patients with FSHD theoretically, but for DMD or tightening, basically exon skipping strategy to increase the expression of the gene is mutation specific. So antisense oligos have to be designed according to the mutation of the patient. So that's why genetic testing is very important, and patient registry is very helpful to start clinical trials.
That's excellent. All right. On that note, I would like to thank you again for taking time to do this FSHD 101 in celebration of World FSHD Day. MDC, as you might know, we're part of the World FSHD Alliance, so we'll be able to share this information with not only Canadians affected by FSHD, but those also in other countries. It's really important that we were able today to raise awareness, to increase the knowledge base on FSHD. And thank you for your ongoing research program that really helps to answer some critical questions. Thank you, Dr. Yokota.
Thank you. Thank you for having me here today.
I appreciate it.
Yeah,
We appreciate
bye.
your
Yeah, yeah,
expertise. On a final note, I want to take a moment to thank Sanofi Genzyme for supporting education initiatives like this, and wish you all a good rest of your day. Happy World FSHD Day, and we look forward to seeing you at our next Let's Talk NMD webinar. Thank you all so much. Take care.